Good afternoon, and thank you very much for attending my talk. Unfortunately, I couldn't be there in person, so this is a recording. I'd like to tell you a little bit about some of the work I did while I was a scientist at the Children's Hospital of Philadelphia. And you may be reading this title and thinking to yourself, what does mitochondrial genetics or genomics have to do with machine learning? Because we don't often associate the short mitochondrial sequence with big data or artificial intelligence. But I'm going to challenge conventional thinking and present to you some of the remarkable complexities underlying mitochondrial genetic analysis. Let's begin. Here's an outline of the talk that I'm going to give today. First, I outlined some of the challenges that we faced in analyzing mitochondrial DNA, including heteroplasmy and new mites. We developed a novel big data machine learning tool called MitoScape to address these challenges, and I'll show you some of the results that we obtained in using MitoScape. First, I'll give an overview of heteroplasmy so that we're all on the same page. I know most of you will be familiar with heteroplasmy, so I'll be very quick in this overview. Here's a cell depicted as a blue circle, and within this cell we have heteroplasmy because we have a mixture of normal mitochondria, shown in green, and dysfunctional mutant mitochondria, shown in orange. As this progenitor cell undergoes cell division, we expect that the mitochondria will be redistributed in the daughter cells randomly or stochastically. What that means is that you can get different percentages of dysfunctional versus normal mitochondria. In this example, all the way on the left, 80% of the mitochondria are dysfunctional, and all the way on the right, you have 100% normal mitochondria in the cell. And in between, you have 60% mutant and 40% mutant. And the idea is only when the amount of dysfunctional mitochondria gets beyond a certain amount or a certain percentage called the threshold do we start to see disease. So why is heteroplasmy a challenge for us? Well, first let's contrast that with the diploid nuclear genome. Since we're dealing with a diploid genome, there are only really three possible outcomes. We could have homozygous reference, heterozygous, and homozygous non-reference. So if we model this statistically, there are just three outcomes to look at. From a statistical point of view, the mitochondrial DNA is significantly more challenging because now we have a polyploid genome. And what that means is now we have continuous outcomes, anywhere from 0 to 100%. And we really need accurate heteroplasmy measurements. To get more accurate heteroplasmy measurements, we need much more data. Here's an outline of the talk that I'm going to give today. First, I outlined some of the challenges that we faced in analyzing mitochondrial DNA, including heteroplasmy and new mites. We developed a novel big data machine learning tool called MitoScape to address these challenges, and I'll show you some of the results that we obtained in using MitoScape. Accurate heteroplasmy measurements are further complicated by the presence of new mites, and I want to spend a little bit of time telling you more about new mites. What are new mites? New mites stand for nuclear encoded mitochondrial sequences. And new mites arose when sequences of mitochondrial DNA transferred into the nuclear chromosomes and got incorporated there. So new mites are actually nuclear encoded sequences, but they're homologous to the mitochondrial DNA because they originated from the mitochondria. These new mites present a significant challenge when you're trying to align sequencing reads to the genome. What you would expect from a typical sequencing experiment is that most of the reads will align to the nuclear chromosome accurately and precisely. And you'll get a lot of the mitochondrial reads aligning to the mitochondrial DNA as you would expect. The new mites, though, are a question mark. The new mites could align to either the mitochondrial DNA or the new mite regions in the nuclear chromosome. Because remember, new mites are homologous to mitochondrial DNA. You can have a similar situation arising where the mitochondrial DNA could align to regions in the nuclear chromosomes corresponding to new mites. So there's this ambiguity here. You don't know exactly where a significant amount of these reads should align. Are they really new mites or are they really mitochondrial DNA? The issue of aligning new mites and mitochondrial DNA is further compounded when you consider the lengths of new mites. New mites can range from 39 bases to almost 15,000 bases. When you consider that the human mitochondrial DNA is 16,569 bases, you realize that a new mite can be almost the entire length of the human mitochondrial DNA. Contrast that to Illumina single reads. The typical Illumina single read today is 150 bases, which is much shorter than the longest new mites. So these are some of the challenges that make aligning new mites and mitochondrial DNA a challenge. When you're trying to distinguish a mitochondrial DNA sequence from its corresponding new mite sequence, one of the things you may think to look at is the base composition. The idea is that some of the bases would be different between the mitochondrial DNA and its corresponding new mite. The problem is that new mites can be 65 to 100% identical in terms of base composition to the mitochondrial DNA. What that means is that it may be impossible to tell whether a short read should align to the mitochondrial DNA or a new mite based simply on sequence composition alone. It is important to realize that new mites are dynamic entities. What this means is that the sequence composition of existing new mites can change throughout a person's lifetime. And even more remarkably, one can also acquire entirely novel new mites during his or her lifetime. One of the contributors to increasing new mites in the genome is stress, and the stress also includes cancer. What this means for our purposes during analysis is that relying solely on a static reference genome can often lead to inaccuracies. So aligning new mites, as you may have guessed from some of the things that I mentioned to you, is not trivial. An Illumina short read that represents your mitochondrial or new mite sequence could align to multiple places. First of all, it could align to the mitochondrial DNA, which means that it could be legitimate mitochondrial DNA sequence, but it could also align to multiple regions in the nuclear DNA. It's not a one-to-one correspondence, as is often assumed to be the case. You could have a single new mite appearing in multiple regions in the nuclear DNA. And there are anywhere between about 755 to 1105 germline new mites. So there are a lot of new mites. A lot of these new mites will occur in multiple locations. There are also some problems with aligning Illumina short reads, including low complexity regions. And a classic example of this is the region between 303 to 317 in human mitochondrial DNA, where you have this long stretch of C's, and embedded in the middle is a lone T there. And there's approximately about 100 GC runs in the mitochondrial DNA. All of these make sequencing challenging, makes alignment challenging. Here's an outline of the talk that I'm going to give today. First, I outlined some of the challenges that we faced in analyzing mitochondrial DNA, including heteroplasmy and new mites. We developed a novel big data machine learning tool called MitoScape to address these challenges, and I'll show you some of the results that we obtained in using MitoScape. So I've told you about some of the problems we faced and some of the issues that we need to deal with when we do the analysis of next-generation sequencing of mitochondrial DNA. I probably don't need to convince this audience of the importance of identifying and quantifying heteroplasmic variants. I want to emphasize, however, that mitochondrial DNA variation also plays a critical role in common complex disease, as myself and other authors have shown. If we are truly interested in diagnosing and understanding human disease through precision medicine, then we cannot afford to ignore mitochondrial variation and heteroplasmy. I also want to emphasize to you that the issues that we're dealing with heteroplasmy and mitochondria are not unique to mitochondria. The concepts and the issues that we're dealing with are very similar to nuclear mosaicism in cells and also cancer. So what do I mean by that? Mosaicism in the cells essentially means that the nuclear DNA in different cells are going to be different. So this could happen throughout your lifetime. You can have different somatic variants in different cells, and this leads to a case of mosaicism. And these situations have also been associated with different diseases and different pathogenicities. And of course, this also happens in cancer. So a lot of the issues, if we can solve them in mitochondria, we can extend that to these other issues in the nuclear genome, and especially cancer. Now to recap, recognize that in order to obtain precise heteroplasmy measurements, we need to accurately identify new mites. Furthermore, we cannot simply rely on sequence composition alone to distinguish new mites from mitochondrial DNA. Here's an outline of the talk that I'm going to give today. First, I outlined some of the challenges that we faced in analyzing mitochondrial DNA, including heteroplasmy and new mites. We developed a novel big data machine learning tool called MitoScape to address these challenges, and I'll show you some of the results that we obtained in using MitoScape. So I talked about a lot of problems and issues. So how did we deal with these problems, and what was our solution for tackling a lot of the challenges in heteroplasmy and new mites? And I want to talk about an algorithm that we developed called MitoScape that used a big data machine learning approach to tackle these problems. Here's an overview of MitoScape, and I'll go into the details of it. The starting point, of course, is a sequencing experiment. Now keep in mind that for the purposes of this work, we only use the Illumina paired-end sequencing, since this is the most common form of sequencing data available. Specifically, we focused on whole genome sequencing studies, and the vast majority of whole genome sequencing studies would have been done on the Illumina platform. Now, in a whole genome sequencing experiment, we start off with total DNA. Total DNA is going to be comprised of both mitochondrial DNA and nuclear DNA. We then perform sequencing on the samples, followed by your standard alignment to the reference genome. This method is a standard approach applied to whole genome sequencing data. There's no difference. Now we expect that most of the reads will align accurately. That is to say, that reads originating from mitochondrial DNA will indeed align to the mitochondrial chromosome. Conversely, reads originating from nuclear chromosomes will accurately align to the nuclear chromosomes. Now, due to the presence of new mites, however, some of the mitochondrial reads will end up aligning to the new mite regions within the nuclear chromosome. Also, some of the nuclear DNA reads corresponding to new mites will align erroneously to the mitochondrial DNA. Both of these cases represent misalignments. So we now need to improve on the first step to accurately identify legitimate mitochondrial DNA sequence. So to tackle this problem, we take the aligned reads and we use a machine learning classifier to further classify these reads. We're using a supervised learning approach, so that means that we have to train the model on known and labeled data, and then we use that model to classify our follow-up data. The difficulty here is how do you get good data that represents a positive training set, which would mean only mitochondrial DNA sequence, and a negative training set, which would mean only nuclear DNA sequence. So to get a database of mitochondrial enriched alignments, what we did was we enriched the mitochondrial DNA from the samples, and then we did Illumina sequencing on it. But we did a more accurate form of sequencing because we use 2 by 300 reads as opposed to 2 by 150 reads to be able to enrich for mitochondrial DNA. And then we sequenced that, and that represented our positive training set for the machine learning classifier. The harder part is actually to get a database of only nuclear reads and no mitochondrial reads. And if you understand mitochondria, that sounds a lot like row zero cells. So you may already know, but for those who don't, row zero cells are cells where the mitochondria is depleted. So you have little to no mitochondria in these cells. And we sequenced the row zero cells that we had, and that gave us a database of only nuclear Illumina reads. So this was a database of mitochondrial depleted alignments. And using these two data sets, we were able to train our machine learning classifier. I also like to leverage the unique attributes of mitochondrial biology and mitochondrial genetics in our machine learning classifier. So if we can use any other information we know about mitochondria and mitochondrial biology to help us classify these reads, that's an advantage for us. So for instance, one of the things that we can leverage is that mitochondrial DNA variants are often in high linkage disequilibrium with each other. In other words, many mitochondrial DNA variants are inherited as a haplotype or a haplogroup. We know of this fact because we know of mitochondrial haplogroups, and we've seen many studies that have talked about associations between common diseases or mitochondrial diseases and mitochondrial haplogroups. So using these linkage disequilibrium scores means that we can compute a probability that two mitochondrial DNA variants appear in the same mitochondrial DNA sequence. If we expect two mitochondrial DNA variants to appear in the same read and they don't, this would increase the probability that the read is actually from a pneumite as opposed to mitochondrial DNA. We can also leverage the fact that from the literature, there are lists of common pneumite loci. And so if a read contains one of these common loci that's associated with a pneumite, then the likelihood of the read belonging to a pneumite is increased. We have a better chance of this read actually being a pneumite as opposed to mitochondrial DNA. And now we integrated both of these sources of information into our machine learning classifier to further classify the mitochondrial DNA reads. And the final output of the machine learning classifier are these accurately identified mitochondrial DNA reads. Here's an outline of the talk that I'm going to give today. First, I outlined some of the challenges that we faced in analyzing mitochondrial DNA, including heteroplasmy and pneumites. We developed a novel big data machine learning tool called MitoScape to address these challenges, and I'll show you some of the results that we obtained in using MitoScape. Now that I've described the algorithm for you, I'd like to talk about some of the results we obtained from running this algorithm on test data. I've described the MitoScape algorithm for you, and now I'm going to describe the formal way that we tested this algorithm. So we started out with human blood DNA samples and performed two separate sequencing experiments on this data. On the left-hand side of the workflow, you'll see that we enriched or amplified the mitochondrial DNA in the samples via PCR amplification. This data set comprises our test or benchmark mitochondrial DNA sequence. We then sequenced this data with 2x300 paired end Illumina sequencing. Now the 2x300 reads gave us more accurate alignments than the typical 2x150 reads. Once we have the sequences, we can call variants on them. These variants are now our benchmark mitochondrial DNA variants. On the right-hand side of the workflow, we perform whole genome sequencing on the blood DNA samples using the standard 2x150 Illumina sequencing. We then take this data and apply MitoScape and call mitochondrial DNA variants. This data set would now correspond to our test mitochondrial DNA variants. And what we want to do now is compare the benchmark mitochondrial DNA variants to the test mitochondrial DNA variants to see how well both of these two data correspond to each other. So we wanted to compare MitoScape to some of the common and popular tools for analyzing mitochondrial DNA from next-generation sequencing data. And those tools fall into two broad categories. The first are unique alignments. So if the read aligns only to mitochondrial DNA, we keep it. Otherwise, we discard it. And an example of that tool is mToolbox. On the flip side, you can obtain your mitochondrial DNA and then do variant calling on it. And then you only do a post-filtering of variants for those variants that we're confident are mitochondrial DNA variants. And an example of a tool that does that is mtServe that I show you here on the right. So in this case, you don't actually get the sequence, you only get the mitochondrial variants. So I wanted to show you some of the comparisons that we did and how MitoScape fared compared to mToolbox and mtServe. So broadly, we classified the types of heteroplasmic variants that we're getting into two categories. The first is low heteroplasmy, so those heteroplasmic variants between 0 to 20% heteroplasmy, and high heteroplasmy, which is between 20 to 50%. And we're looking at false positives and false negatives. False negatives would be those variants that the tool missed. False positives would be variants that the tool called but were not actually real variants. And you can see clearly for the low heteroplasmic variants, MitoScape did not have any false negatives or false positive. The worst tool in this category was mToolbox. There were 81 false negatives. mtServe did a lot better. There was only one, but it also had a lot of false positives, 3 false positives for mToolbox. So at least for low heteroplasmic variants, MitoScape was the best tool, mToolbox was by far the worst. You would think for high heteroplasmic variants that the tools would do a lot better, and you should accurately and precisely get all of the high heteroplasmic variants. But surprisingly, even MitoScape had a false negative for the high heteroplasmic variants. But you can see from this data and these results that MitoScape is still doing significantly better than mToolbox and mtServe. So one of the important metrics is the predicted heteroplasmy level for each one of these tools. And we can get a measure of the error in this heteroplasmy prediction by taking the difference of the predicted heteroplasmy minus the actual or known heteroplasmy for each individual variant and for each tool. And that's what I have plotted on this graph here. So on the y-axis, we have the scale heteroplasmy error. So we'd expect that high heteroplasmy values will also have high variances. So we needed to account for the actual heteroplasmy value by scaling by that measure that you see here. And on the x-axis, we have the read depth. So ideally, we would want the difference in the heteroplasmy values or the error to be 0. And you can see quite well here that the average heteroplasmy error for MitoScape was a 0 and remains 0 regardless of the read depth. There is some variability in the amount of error because you see the difference between the dashed lines is not 0, but it's much smaller than it is for mToolbox and mtServe. In the case of mToolbox, that variance in error only started really decreasing until we got past 800 or even 1,000 in read depth. So you really needed high read depth for mToolbox to give you accurate heteroplasmy levels. mtServe, on the other hand, didn't do very well at very low read depths, below 200, but it quickly improved as we got past 200 and again past 600. But overall, you can clearly see that MitoScape is predicting much better values for heteroplasmy levels compared to the other two common popular tools that we see. So everything that I presented to you was published a couple of years ago in PLOS Computational Biology, and I would direct you to this publication in case you have further questions. I trust that you can appreciate that mitochondrial DNA heteroplasmy is an important challenge, and MitoScape addresses many of these challenges and issues by applying big data analysis and machine learning together with mitochondrial biology and genetics. MitoScape was designed to be scalable and undergo iterative improvement. So as we get more mitochondrial DNA enriched data, more row 0 cells, and we learn more about mitochondrial variants and new mites, we can improve the model and improve the variants that we're calling with MitoScape. So thank you very much for your attention, and I'll take any questions now. Here's an outline of the talk that I'm going to give today. First, I outlined some of the challenges that we faced in analyzing mitochondrial DNA, including heteroplasmy and new mites. We developed a novel big data machine learning tool called MitoScape to address these challenges, and I'll show you some of the results that we obtained in using MitoScape.